Anyway, with that said, I'm sure all my recordings start too with me going anyway. But here we are. So let's go ahead and pull up the PowerPoints for chapter 14. Yes. Okay. Ooh, is it going to be? Okay. Huh. Let's see if we'll see how this works out. Can you guys see that? Oh, my screen sharing is paused. Okay, so no, you can't. Let me move this back down here. Huh. Well, this is going to be interesting. What happens if I move this up here? Okay, and now can I move this up here too? No. Yes, yes. Can you guys see that? Nope, 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 never mind. You can't. Okay. But I can move this up here. Okay. So what I can do is that I can move this up here too. Yay, technology. Okay. And I can find a way to resize this. Come on. Oh my Lord. Why is that doing that? You can laugh at me for my awful technology. All right. All right, let's put this in slideshow mode. There we go. Okay. Now it's funny. Ah, I see that. Okay. Everything good? You guys can see this?
Yeah, we can see it.
Okay, awesome. Good, good. Because when I share it that way, it pops up on my other screen. All right, but now I know. Okay, so we kind of looked at this last Wednesday, but we're going to go through glycolysis and gluconeogenesis today. And then on Wednesday, maybe do a little discussion about all this as well. So I'm thinking our first exam will probably be 2 weeks from today. And that'll give you plenty of time. Maybe on the Wednesday before, but we'll be ready for it the Wednesday before, but we'll probably push it 2 weeks from today. So that'd be February 29th or March 1st. Okay. But we looked at the glycolytic pathway, and we actually have 2 parts of it. The first part is the prep phase or the investment phase, where we spend a couple ATPs, and then we let's see here. You gotta click down here, don't I? No, maybe. Yeah. Oh. Come on. There we go. This way. I see. I've got it covered up down there. Okay. And then we have the payoff phase where we start producing ATPs, okay, as well as NADHs. Okay. And this is where we're going to get our energy back out of glycolysis. So we spend 2 ATPs, we get out 4 ATPs and 2 NADHs. And the key is we're taking our 6-membered glucose and cleaving it into 2 3-membered glyceraldehyde 3-phosphates. And then as we phosphorylate that and we start making our ATPs, our NADHs. Okay, so let's talk about all these reactions in excruciating detail. Okay, and I tell you, this little thing here is going to be in the way no matter where I put it, isn't it? Okay. So the first thing that happens, our very first step, is I drink my Mountain Dew here, and I can't right. Yes, thank you. I drink my Mountain Dew. All right. And I get my glucose in my bloodstream, and it gets into the cells, the endothelial cells in the lumen of the gut, and hexokinase binds glucose and phosphorylates it at position 6. So I make glucose 6-phosphate. This is a very favorable reaction. So we can see here this happens pretty much spontaneously, but it is also only going to go one way. We're not going to spontaneously dephosphorylate this. My analogy here as well too is because our glucose transporters, you might remember from Biochem 1 that the glucose transporters kind of will transport them in or out of the cell. So when we bring them in and phosphorylate them, that keeps them in and they can't get back out. So it's like the crazy cat lady who puts the little tags on the cats so they can't get outside once they get in her house. So Hexokinase, oops, gotta remember how to do this. Our hexokinase is trapping that glucose inside the cell and is basically lowering the intracellular glucose concentration so we can further uptake it. So once I convert glucose inside the cell into glucose 6-phosphate, it's different, so I can pull more glucose in. So as I phosphorylate this, I can bring in more glucose. So I can absorb all the lovely, lovely sugar in my bad habit drink. I wish I'd been a coffee drinker. Okay, let's try doing this way. There we go. Okay, just to see. Okay, good. Okay, things working. All right. This requires ATP. So of course we don't want it to be irreversible because we don't want to waste our ATP. The other neat thing is we have lots of different flavors of hexokinase. We got several isoforms. Okay, so 1, 2, 3, and 4. We generally talk about hexokinase 4 as glucokinase, but different organs are going to have different hexokinases in there to deal with the differing concentrations of glucose. You think like your muscles, I think have muscles and liver might have hexokinase 2, lumen has hexokinase 4. I had to look up some of the others in there. All right. Anyway, we have this lovely little nucleophilic attack here, attacking that phosphate of ATP. And of course, with that magnesium in there, it's going to facilitate this process, shielding some of those negative charges on there. Very thermodynamically favorable, so it's effectively irreversible. Okay, so this is our first regulated step. Okay, and it's mainly regulated by substrate inhibition. If we have lots and lots of substrate, then we don't do this as much. Okay, so we have lots of glucose 6-phosphate, lots of ATP, we won't see this as much. If I move to our second step, one day I'll remember. I got to retrain myself here. Now we're going to isomerize this and make fructose 6-phosphate. Okay, so essentially we're going from a 6-membered ring to a 5-membered ring. Okay, so we're not really losing anything or adding anything. We're just rearranging. And what this does is this allows us to have this other little nicely accessible hydroxyl group sitting out here on carbon 1. Okay, you can see in the pure solution energetically, not terribly favorable. It's actually slightly unfavorable. But if we have lots and lots of glucose 6-phosphate, we're going to push that from the reactants to the products. So this is reversible. Okay. So if I have lots of fructose 6-phosphate, I'll isomerize back into glucose 6-phosphate. Okay. But for glycolysis, we're going to proceed forward here. We're going to drive this forward. And then we see how this gets driven forward, because once we have fructose 6-phosphate, oops, okay, sorry. I forgot the thing I'm going to talk about here. We'll get to that in a second. But it makes the next steps easier. Okay, so by rearranging that, it's a lot easier for that fructose hydroxyl group to interact with the next step in here, basically to be phosphorylated. So basically our aldose, this is our keto aldo form. We have this diol intermediate. So we've probably seen that in various places before, but we'll take a look at that here on the next slide. And it is reversible. So if I look at this next slide here, here's what happens. If you recall with glucose, when glucose is in solution, it can form a ring structure and it can linearize as well too. And this is constantly happening. This is why we have that alpha carbon in there. So we're constantly opening up there. So in this case, our phosphohexase isomerase, once this opens, we have this basically this base on the enzyme. I swear, I turn off that phone and it just turns back on. Anyway, our base kind of attacks this proton here on carbon 2, which is going to kind of flip this up this way. Okay, so now we have formed this ene intermediate in here. Okay, and now we have 2 alcohols here. So this is our ene diol. So we're going to kind of flip this one down here and move this up here. So we have some acid-base catalysis. We get our proton back. Okay, but we've now moved this into this position like so. And now when we make a ring structure, okay, we actually end up with a 5-membered ring. And this is now out here. So you can see our carbon 1 is our hydroxyl group on that carbon 1 is now more accessible. Okay, so we've essentially kind of switched places between our carbonyl bond and our carbon alcohol bond there. Okay, that enediol. If we were going into excruciating detail, we talk about this mechanism and so on and so forth. But the key here is our isomerase is actually interacting with and abstracting a proton for a moment and then adding it back. So you can see we take away a proton and then we add we add a proton and we take away a proton. All right. Okay. But we end up with this nice little structure here. Okay. All right. And then this goes into the mechanism in much more detail than we really need. Okay. Okay, our third step, once we have our fructose 6 phosphate, PFK1, phosphofructokinase 1, is going to phosphorylate carbon 1 so that we make fructose 1,6 bisphosphate. So now we have two phosphate groups on here. Again, we're going to use ATP, okay, as our source. Again, just like before, our glucose, when it had this kind of exposed hydroxyl group here on carbon 6, that made a good nucleophilic attack on ATP. Our newly exposed hydroxyl group here on carbon 1 will do the same thing with ATP. So we'll phosphorylate that. And again, you can see very favorable, unidirectional. This is effectively irreversible. Okay. So what we're doing by spending this ATP to phosphorylate glucose twice, we're activating it to make it more, we're prepping it, we're priming it. It's like all the training you do before the big game. I don't know, something in there. The other thing is making our fructose 1,6 bisphosphate. This is the first step of glycolysis. F16BP is only going to go in a glycolytic pathway. So glucose 6-phosphate has several different fates. It can go down several other pathways, but now we're like, okay, here you go. We're going for glycolysis. Okay. Again, irreversible. PFK1 is very highly regulated. So ATP, fructose 2,6 bisphosphate, and many other metabolites will actually bind to the enzyme and regulate it. So if we have high ATP levels, it's basically going to downregulate it. Okay, so we already have lots of ATP. We don't need to make more energy. Go do something else. Like I tell my kid here, it's like, I'm teaching right now. Go do something else. Okay, so essentially, PFK1 says don't burn glucose if we have plenty of energy. We can store it as glycogen in the liver or use it to build other components with. But once we make fructose 1,6 bisphosphate, okay, we are going to proceed through glycolysis. Okay, so the good news is the next several steps are unregulated. They're just going to go forward here. So our fructose 1,6 phosphate, the aldolase will interact on that and cleave between carbon 3 and carbon 4 so that we make dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, so we essentially now make 2 3-carbon units that are each phosphorylated. Okay, you can see this is not energetically favorable. But if we have lots of fructose 1,6 bisphosphate, we're going to drive that forward, especially if we use up our products as we make them. Okay, so these are high energy phosphate sugars. Our reverse process is an aldol condensation. So if we have lots of DHAP and G3P, it will readily condense to make our fructose 1,6 bisphosphate. Okay. There's really kind of a neat study of the mechanisms in here. Animal and plant aldolases use covalent catalysis, and funguses and bacterial aldolases will use a metal ion catalysis. So, two different mechanisms to do the same thing. So this might be an example of convergent evolution. That's basically trivia for us at this point. But again, this is unfavorable, very reversible. So what we do is we make that glyceraldehyde phosphate very low concentration to pull it forward. Okay, we don't need to add energy in there. We just need to make sure there's very low levels of these products. Okay, so when we make our glyceraldehyde phosphate and our dihydroxyacetone, what we really want some mechanism. This is I don't know. I tend to gloss over this because there's so much information, but it is kind of neat to review this because you can see your organic chemistry happening here. We get our Schiff bases that are protonated. We can go down through here and we eventually release these, and we have proton exchange to restore the enzyme as well too. Okay, but anyway, we're not going to worry too much about that. That's just the mechanism of how that works. All right, so we want glyceraldehyde 3-phosphate. That's awesome, that's great. But we have, yeah, okay, sorry, too much information here. Okay, stop, okay, there we go. But we also make this dihydroxyacetone phosphate. Now again, you can see here we have this, kind of a ketone. So we can go an enediol intermediate, much like we did before. And we have another enzyme in there called triose phosphate isomerase that's going to rearrange this to put make this into an aldehyde as opposed to a ketone. Okay, and when it does that, our dihydroxyacetone phosphate gets converted into a second molecule of glyceraldehyde 3-phosphate. Again, this is not super favorable, but as long as we're using up our glyceraldehyde 3-phosphate, this will keep going. Okay, so at this point, we have now made 2 molecules of glyceraldehyde 3-phosphate, and we need the glyceraldehyde 3-phosphate because it's our substrate for our 6th enzyme for our step 6. Okay. Once we interconvert our DHAP into our G3P or GAP, different abbreviations, then we are ready to go into our payoff phase. Okay, so if we take a look at this, if we take a look at this closely, we can actually look at where our different glucose carbons end up. Okay, and we can see here, we cleave like 4, 5, and 6 immediately go into our glyceraldehyde 3-phosphate, and then these make our DHAP, which get interconverted into G3P. Okay, in the past, sometimes I've made challenging questions. I think you might recall from Biochem 1, suppose there's a world where instead of hydroxyl groups, you have sulfhydryl groups. What product do you make when you do some type of reaction in here? Still turns out to be water because the sulfurs actually stay in the compound. But it's always tempting to figure out which carbons are going where. Okay, same thing with the phosphates. So you could use radio-labeled carbons, radio-labeled phosphates to figure out, where these particular atoms are going, which can help understand the mechanism. Okay, but again, that's just peeking below layers more than we need. What you guys need to know is that we cleave our fructose 1,6 phosphate into DHAP and G3P, and then we convert that DHAP into G3P, and then we're ready to make energy. We're ready. Our investment is maturing. Our berry bushes or fruit trees have grown and are producing. Okay, so next we're going to start oxidizing. All right, so we have our glyceraldehyde 3-phosphate. We're going to take inorganic phosphate. Okay, so we're basically going to add another phosphate group here to our aldehyde end. And when we do this, we're going to see this lovely little oxidation, this redox reaction. We're going to produce NADH. Awesome. NADH is our high energy molecule. You'll recall in the electron transport chain, it plays a pretty significant role in our complex 2, I believe, and succinate dehydrogenase. So we're essentially getting effective energy units out by making NADH. Okay, and keep in mind we have 2 of these now, so we're getting 2 NADHs out. Okay, and we make this 1,3-bisphosphoglycerate. Okay, so 2 phosphate groups on here. Again, not a super favorable reaction, but as long as we use up our 1,3-BPG, we will be pulling it forward. Okay, the neat thing here is this 1,3-BPG is a high energy phosphate compound. Okay, so we're not using ATP, we're using inorganic phosphate floating around in there. And the neat thing is by making this high energy compound, we can actually make ATP in an upcoming step. Okay, our NADH is our first step here. Our active site is a cysteine, so we're going to have this thioester intermediate. So oxidative stress will inactivate this, but that's more than we need to know. Let's just, for fun, take a look at this. Okay, so here's our glyceraldehyde 3-phosphate. Our NAD plus is bound here. We have this cysteine residue, which is going to actually bind to our carbon here. Okay, so now we have a tetra valent carbon in here. Um, our hydrogen is going to get abstracted off of here onto NAD plus to make NADH, bringing with it its electrons as well too. So now we end up with this nice little compound here. All right, we bring in some inorganic phosphate. Okay, and as our inorganic phosphate comes in here, we're going to see this nucleophilic attack to the carbon, which is going to kick our sulfur back off, regenerate our enzyme. We've now phosphorylated here at this position to make our 1,3-BPG. Okay, good times, right? I always like this because tracing where these electrons go, I think helps you understand what's going on within the chemistry that's in there. Okay, and maybe helps solidify the rationale for how these reactions are occurring. All right, substrate binding. Okay, analysis. Okay, all right. So now that we have this 1,3-BPG with this high energy, phosphorylated 3-carbon sugar, we can start making ATP. We're going to directly transfer this to ADP. All right. So we basically pull off our phosphate group that we just added and put it on ATP. So we have that little 2-step process in there to make the energy. And you can see this is a very favorable reaction. Yay! So phosphorylating ADP is actually very favorable because 1,3-BPG is a very high energy intermediate. Okay. If you go back to that chart in chapter 13, you'll see 1,3-BPG up there with like creatine, phosphate, and all that good stuff. All right. This is a substrate level phosphorylation. That's what we call that. We're basically just transferring that phosphate group in there. The kinases are ones that transfer phosphate groups between ADP and various substrates. So we have this phosphoglycerate kinase transferring the phosphate from the 1,3-BPG to the ATP. Very favorable, and it's very reversible. Okay, we can reverse it and go the other way too. We can use ATP to phosphorylate that. And again, as long as we keep our 1,3-BPG levels low, that would occur. Okay, so now what we have left over is 3-phosphoglycerate. So we're going to move this and make 2-phosphoglycerate. Okay, so we have this mutase here. We could just as well call this an isomerase, but it's named a mutase, and we're essentially going to place this on position 2 here. Just to make our second high energy compound. Okay. All right. So this is our second high energy phosphate compound. All right. We have an active site histidine. So we're going to make this a phosphohistidine in there, and it's going to donate that phosphate group to carbon 2, and then it basically gets the phosphate from carbon 3. So what we're doing, we're not really taking this phosphate and moving it over here. We're taking this phosphate and putting it on the mutase, which has a phosphate, which releases its phosphate to position 2. So we're just kind of trading them. Go through the whole Indiana Jones and the little bag of sand and the idol. He's just trying to gauge them and move them over at the same time. So we've got 2 different phosphates. One is on the enzyme. Okay. This is not really a very favorable reaction, slightly unfavorable, but it's reversible. So again, as long as we keep using up our products, we can keep pulling it forward. Okay. All right. No, and here's our description of the mechanism that's occurring. Fun stuff, right? Okay. All right. Acid-base catalysis in there. Okay. So finally, what we have now is our 2-phosphoglycerate. I'd say almost finally. What we're going to end up doing now is having a dehydration reaction. Okay, so we're going to lose water here, and we're going to end up making this ene, basically this enol. Okay, so we get this phosphoenol pyruvate. Okay, so enolase is going to catalyze the dehydration of 2-phosphoglycerate to phosphoenol pyruvate. And PEP, very high energy compound. Once we have PEP, we will have enough energy to make our second set of ATPs. Okay. Rationale for this is our 2 negative charges in 2-phosphoglycerate are very close together. So what we need to do is stabilize this, okay? And again, slightly unfavorable. So finally, we have phosphoenol pyruvate, okay? So it's going to be acted on by pyruvate kinase. Again, we know our kinases transfer phosphate groups. So it's going to transfer the phosphate group from phosphoenol pyruvate to ADP to make our ATP. And you can see, boom, this is a very favorable reaction to the point where it's irreversible. Okay, so this is our very last step in glycolysis. It is going to be highly regulated. Okay, um, so again, this is our second substrate level phosphorylation. We get an enol that tautomerizes into a ketone. This is going to lower the concentration of the reaction product. So again, we can continue to drive this forward to making ATP. Okay. We got to have magnesium or maybe manganese for activity, depending on the species and organ. Very favorable, very highly regulated. Again, if we have lots of ATP, it's going to slow it down. If we have our divalent metals like magnesium or manganese, that may speed it up. Various other metabolites will regulate this as well too. We'll talk more about that regulation in chapter 15. Okay, and here's our tautomerization. Okay, so our enol form and our keto form. Yay. We typically tend to be in our keto form. So it's like everybody's on the cool keto diet now. Nobody's on the enol diet. Okay, no, sorry. It's bad jokes. All right. So those are the 10 steps. And like, oh my Lord, do I have to know this? Yep, mostly. It's good to do it, but I'm not going to make you memorize every single step in here. But you should know the overall component. Remember way back in general chemistry when you had to do like the complete ionic equations and all that? Well, here we're looking at our net equation. We take one glucose plus two NAD pluses plus two ADPs plus two inorganic phosphates. And from that, we get two pyruvates, two NADHs, two protons, and two ATPs. Okay, so we use up this and we produce this. So this is why we get two net ATPs. All right. Again, glycolysis is very heavily regulated. Okay, and the reason for this is we want to make sure that as an organism that we are in the properly fed state, that we have access to nutrients when we need them. Okay, so if I eat a big meal, I've got lots of nutrients kind of in my gut floating around, so I want to store those for later. If I haven't eaten in a while, I want to release those from storage so that I can use them. So we make our ATP when we need it. Okay. Later on, we will talk about some cases where fight or flight. When you get surprised, a bear jumps out of the woods or something like that. Clown comes out of the sewer with a red balloon. I don't know. So if you need ATP right away, we'll see some of these reactions that will very quickly trigger the rapid release of energy, and that will include glucose and upregulate glycolysis so that you can make lots of ATP so that you can fight or flee. Okay, let me pause for a second and pause for station identification. If you guys have questions about any of this that we've gone through, it's a good time. Otherwise, we'll jump ahead into the logic behind this. Okay. All right. So there is a logic, and apparently I do enjoy music, even if I don't have much of a musical talent, because there is an inner logic making me think of like Bad Religion songs from the 80s or early 90s. Oh my Lord, old. Anyway, our chemical logic here is that we are utilizing the carbons in glucose and redox reactions so that we can make pyruvate and energy. So, we're moving all this around so we can get higher energy stuff going on. Okay, so we can make our ATPs and we're left with pyruvate. Okay. All right. Sometimes I think going through these pathways can be helpful. Sometimes maybe they can be a little confusing. I kind of leave it in here up for you to see if you like it. That's good. If not, you can maybe ignore this slide. But this is just basically a nice little summary of what's going on in here and the various chemistry that's occurring in there. Okay. Okay. So what all feeds into glycolysis? Okay. Well, we get our glucose from our diet. Woo, yay, a really bad diet of Mountain Dew. Or we can actually, if we're not consuming Mountain Dew and go out and run a marathon or something like that, glycogen can be released it, or here a plant starch. But we have glycogen phosphorylase that will cleave a glycogen from the stores, cleave glucose from the glycogen stores. It gives you glucose 1-phosphate. So we will rearrange that and make our glucose, our fructose 1,6-bisphosphatase. Our disaccharides can be hydrolyzed. So if we drink our milk this morning, lactose, we get glucose and galactose. Sucrose, have your glucose and fructose. Neat thing is fructose is already in a form. That's useful there too. We have a variety of monosaccharides as well too. But all of these can enter into glycolysis at different points. So you can see our sucrose produces glucose, so that can be, of course, phosphorylated. Our fructose can go directly to the fructose 6-phosphate. It can be phosphorylated as well too. We don't need to do that rearrangement in here. Mannose, sugar of fate, but it can be rearranged. Again, we see an isomerase in here to convert mannose 6-phosphate into fructose 6-phosphate. Galactose can be converted into glucose, our UDP glucose. We cleave off the UDP and add on a phosphate group, and we're ready to roll. Okay. All right, you can also see fructose has an alternate pathway. Fructose can be cleaved into glyceraldehyde and dihydroxyacetone phosphate as well too. So there's our fructose 1-phosphate. Okay, so if we make fructose 1-phosphate instead of fructose 6-phosphate, we can kind of skip the step here, but that's just an alternate pathway. Trehalose, lactose, all sorts of stuff in here. You can see all these different enzymes that are involved. Glycogen or amylases. So like this dietary glycogen versus endogenous glycogen. All right, anyway, so at the end of this, we've made some ATP. Yay, our body's happy, we have energy, but we also have made pyruvate. So what happens to the pyruvate? So the fates of pyruvate become actually fairly important in here. We have to learn where they go, what happens to them. So under hypoxic conditions or anaerobic conditions, we see this with, like, say, yeast. Our pyruvate can be converted to ethanol and carbon dioxide. It's kind of funny. My graduate career, I worked with yeast. I worked with a lot of yeast. I would grow up like 24 to 40 liters of yeast at a time. So it smelled a little bit like, baking bread or beer, but it also smelled like like stuff growing. So it was kind of simultaneously maybe a little bit hungry and a little bit sick. But anyway, that's under anaerobic conditions that we can make ethanol there. Okay. We could also make lactate. So we typically see this in yeast. In us, if we exercise, go running or something like that, our muscles are going to, become starved for oxygen after some time. So we'll make lactic acid. Lactic acid will build up. That's where you feel the burn and all that good stuff in there as well too. And then you can bring that back to the liver and have it reconverted back into pyruvate. But anyway, lactic acid is another fate of pyruvate. And the other thing we can do is we can convert under aerobic conditions. So we've got oxygen available. We can make acetyl-CoA from our pyruvates. And our acetyl-CoA will feed into our citric acid cycle. Hey, look, it's chapter 16 material. Okay, so we can see where we're going to go with that in there as well too. And then eventually we'll make carbon dioxides and water in here. Okay, all right. So so pyruvate has three major roles three major fates. Well, actually 4, because it can get converted back into glucose too, but more on that in a moment. Okay. Just very briefly talking about fermentation. We're going to generate energy without consuming oxygen or NAD+. So we don't see a change in the oxidation state. No oxygen, anaerobic, no oxygen, no change in oxidation state. So we're going to do this to produce beer, yogurt, soy sauce, all sorts of stuff in there. We'll have a fermentation that goes on, probably kimchi to some extent. For us and other animals, we will make lactic acid. Okay. Not right now. In 20 minutes. Go.
No. I'm going to do that right now, and I don't know
20 minutes and I'll be right there. Okay, go.
20 minutes is 10 hours.
Yeah. Okay. But anyway, our lactic acid builds up, and oh my goodness. Just as an aside, I remember when my son is the older child, when he was little, when he was 3, I could bring him to class and he would just sit there so quietly and so nice. He would never know he was there. This one needs lots and lots and lots and lots and lots of attention. Anyway, lactic acid, if we let that build up continuously, we get basically acidification of our muscles, which means we can't continually work out. That's why I always think back to when I was a freshman and we had a rock climbing course. So, I got the nickname Spider-Man because I could climb everything except for this 60-foot thing that had like a 5 or 6 foot overhang. I was a skinny little kid with no muscle tone. I got up the rock, but trying to go over that way, I could actually feel my muscles were actually shaking like this because I had burned so much energy in there that the acidification of my muscles, eventually kind of couldn't hold on and got to fall and swing just a little bit. But anyway, what happens if you can't have that, if you get that lactic acid buildup, which is why elite athletes will look at ways to remove lactic acid. You might have seen like years ago, erythropoietin was a big doping concern because it helps transport out lactic acid. So all sorts. Well, anyway, we're getting off topic here. Thinking about this is we need this recovery time. Okay, so there is kind of a recovery time to remove the lactic acid, reconvert it back into pyruvate or glucose in the liver. Okay, all right. If we look at this is the way it works. Lactate dehydrogenase, very abundant protein. In Biochem lab, I used to get a beef heart from one of the local butchers, bring it in and have students over the course of several weeks purify lactate dehydrogenase from the heart muscle. It's actually fairly easy to do, unless, of course, your refrigerators keep breaking down or you keep clogging your columns. But anyway, this is the reaction that goes in here. We make lactic acid. You can see that this is a very favorable reaction. So our glucose goes to pyruvate to lactate here, and you can see we have our NADH involved in this as well. Okay. Oh, pardon me. Okay. So let's switch gears a little bit. Talk about glycolysis. In tumor cells, because they are fast growing, pretty much in any fast growing cell, we see elevated rates of glycolysis. Okay, so here we have our cancer cells tend to upregulate hexokinase as well as our glucose transporters, so they can bring in more glucose. If they're bringing in more glucose, they can undergo more glycolysis. If you look at some certain anti-cancer agents, they inhibit the phosphorylation of glucose. So like our bromopyruvate, our monoamine will all bind to our hexokinase and prevent this phosphorylation in here. We have another anti-cancer agent, oncogenic drug, that will inhibit this reaction as well too. But anyway, we upregulate cancer cells because they're fast growing, need lots of energy, so they upregulate glycolysis, so they can produce all this energy in here, so that you can have increased cancer growth as well. All right. So tumor cells often have high levels of lactate dehydrogenase. So rather than beef heart, we could use a tumor. That would not be a good lab. All right. Our yeast in here, we're going to make ethanol. Neat thing is, you may have seen, we don't have pyruvate decarboxylase, but we do occasionally have yeast that are symbiotic with us. There are some people who will produce ethanol naturally through a yeast colonization, either in their throat, so like thrush, or sometimes people will have, there are instances of people who will have yeast growing in their gut somewhere as well too, and they can actually produce ethanol that way. So anyway, this is basically what our fermentation process looks like here. You have our pyruvate goes to acetaldehyde to ethanol. Okay. Um, yeah, so, and of course, this is reversible. Okay, so alcohol dehydrogenase will, if you drink lots of ethanol, your alcohol dehydrogenase in your body will convert it to acetaldehyde, and it can be converted to pyruvate, or it can be used, as a ketone body for fuel. The issue that you would have with drinking wood alcohol or methanol is that alcohol dehydrogenase will also bind methanol. So there we just have a one carbon alcohol, and it does pretty much the same reaction, except we don't have this methyl group here. So instead of forming acetaldehyde, we form formaldehyde. Okay? And when we make formaldehyde, what do you use formaldehyde for? As a preserving agent for smells like med student. That's what like if you ever did fetal pigs in high school, they were packed in formaldehyde. The cadavers in medical schools are often packed in formaldehyde. Formaldehyde is a great preservative. It's also pretty nasty poison. It tends to accumulate in the optic nerve. So if you drink too much bad alcohol, you can go back to prohibition for this. Talked about getting pickled. That'd be one of those things making formaldehyde because quality control wasn't great, and there was a lot of methanol in the ethanol that was produced by bootleggers. Same thing, people would go blind from drinking bad hooch because they'd get that methanol in there that turn into formaldehyde and kill the optic nerve. So quality control is good, but to go back, kind of review a little bit of enzyme kinetics. We have ethanol. What happens if you are, say, on a boat, 100 miles offshore, and you reach for a bottle of tequila, and you accidentally get a bottle of rubbing alcohol. Well, actually, that's not rubbing, wood alcohol, and you drink a whole bunch of methanol, and you're far from a hospital. Any suggestions on what you can do to, kind of maybe help stave off the formation of formaldehyde by your body? So probably one thing is induce vomiting, get anything out of your stomach. That's in there as well too. Um, activated charcoal tablets would help as well too. And then the other thing you do is actually chug that bottle of tequila. Okay, that's bad, but that would be, you could compete to slow down. So if you have ethanol and methanol both competing for binding spots in here, that can help until you can get closer to medical treatment. Anyway, things to know. Don't drink methanol. All right, but anyway, ethanol fermentation there. Okay, so this leads us into our talk about TPP. I want to show my age and quiz you guys. Are you guys down with TPP? Because 90s rap videos are stuck in my head. But our TPP is a very common. This is our thiamine pyrophosphate. We'll see this. It has this lovely little thiazolium ring in here, and this is a superb acetaldehyde carrier. Okay, so we can actually carry this nice little group on here, and send this through so that we can basically have this aldehyde group that can be eliminated and transferred somewhere. So basically, we can attach an aldehyde one place and remove it someplace else. Okay, so we'll also see TPP as being an important cofactor and participant in some reactions as we go through the semester in here. Okay. All right, so common acetaldehyde carrier, thiamine pyrophosphate. This is its, not going to make you guys remember the total structures or draw these structures. That's a little bit too much. But we can see some TPP-dependent reactions, pyruvate decarboxylase for ethanol fermentation. But we're going to look here at synthesis of acetyl-CoA, right? And we're going to look at carbon skeleton reactions in the pentose phosphate pathway. All of these require thiamine pyrophosphate. If you think thiamine, you're like, wait a minute, isn't that like a vitamin, B vitamin? Yes. So that's why our B vitamins tend to be very important. All right. Enough of that, right? We'll see more on TPP in a little bit. For the remainder of the class, go backwards. All right. What we've got here is gluconeogenesis. So if I drink my Mountain Dew, I have lots of glucose in my bloodstream. I can use that to make energy. Okay, but suppose I don't drink my Mountain Dew, I don't eat anything before lunch, and hold on a second, I got loud children in the background. Be right back. Okay, sorry about that. I find I get distracted easily by noisy environments. Anyway, um, so if I need more energy, if I need more glucose in my muscles, I'm working out here and I don't have anything circulating in there, I can actually make new glucose in the in my body. Okay, so I can't convert fatty acids to sugars, but I can do something else. I can take my lactate, and convert that into pyruvate, feed that into the citric acid cycle. I can have breakdowns on various amino acids. And all these can be used to make um, glucose that I can use for energy. Okay, so let's take a look at how this happens. It's 70% the same as glycolysis because all those reversible reactions in there, we're just going to go the opposite way. So instead of converting 1,3-bisphosphoglycerate into 3-phosphoglycerate, well, I'm going to go the opposite direction. And you can see here, instead of making 2 ATPs, I'm going to spin 2 ATPs. So I am investing energy to make my glucose in here. We have 3 enzymes that are different, okay? And you can see these kind of like our roundabouts here, okay? So 7 of them are the same. 3 of them, we need different enzymes because these are the irreversible reactions. Okay? Keep in mind our glycolysis is primarily in the muscle and the brain, gluconeogenesis mainly in the liver, so that our liver will release that glucose so it can go to the muscle and the brain and provide the energy for ATP production in the muscle and the brain. Okay, so gluconeogenesis, we're going to start with pyruvate, and we have a little 2-step process to make phosphoenolpyruvate, and then everything's the same until we get up here to our fructose 1,6-bisphosphate. We have a phosphatase. Phosphatases remove phosphate. Okay, so make our fructose 1-phosphate, and we have another phosphatase over here that does the opposite reaction of the hexokinase. Okay, so our phosphatases and kinases are kind of at opposite ends. Our kinases are adding phosphates. Our phosphatases are removing them. Okay, and here you can see our kinase, and we have our carboxylase and our carboxykinase. Okay, so let's look at this. Both pathways are favorable, but they go in opposite directions. So we need regulation to make sure that we're not just burning energy for no reason. I make pyruvate and immediately convert it back into glucose. So that's not going to be good. So our irreversible reactions are the ones we focus on because there are bypasses. Okay, so we don't make ATP during gluconeogenesis. Okay. We actually have to spin some ATP to make our glucose, but then we can send it to the muscles or the brain where we can make ATP. But gluconeogenesis itself does not make ATP. All right, and we have differential regulation so that we don't have that futile cycle. So if I activate glycolysis, I want to make sure I'm inhibiting gluconeogenesis, and vice versa. If I activate gluconeogenesis, I'm going to inhibit glycolysis. So we strive for efficiency. So let's look at these steps. Steps 1 and 2, we have this little 2-step process in here. We're going to convert pyruvate to phosphoenolpyruvate, but we can't go directly there. That just is too difficult. So we're going to take 2 steps to do that. The first one, we have this pyruvate carboxylase, which is going to convert pyruvate to oxaloacetate. This is going to involve using biotin as a cofactor. And we're also going to have to be in the mitochondria to do this. So we're going to do some compartmentalization to separate out. Okay. Once we have our oxaloacetate, we're going to convert that to phosphoenolpyruvate. All right. This occurs in mitochondria. It can occur in a cytosol depending on the organism, but you can see here we're moving stuff into the mitochondria. We're going to do this in the mitochondria and then move it back into the this is kind of a neat thing that's happening here. We're going to add a carbon and immediately remove that carbon from the structure. Okay, so that's why we have this biotin involved as a cofactor in here to add it and then take it away. Okay, so here's our bicarbonate, here's our pyruvate. We essentially are adding this carbon here to get our oxaloacetate. So we go from a 3-carbon compound to a 4-carbon compound. Okay, all right, so here's what we have happening. Okay, the neat thing is biotin is this kind of long chain, so it can move around a lot. So it can take our methyl group from one place and move it around into its proper position. So that's kind of what's happening here. We have this initial site and this second site. So we're basically kind of attach this bicarbonate to biotin at one position. It's going to rotate over into the second position and transfer that carbon immediately to pyruvate to make oxaloacetate. Okay, so put it on over here, take it off over here. So if you think of like the old bucket brigade, I mean, here's a bucket of water. I'm going to pass it to the next person. I'm going to come back and get another one, pass it to the next person, on and on. So that's essentially what our biotin is doing here in pyruvate carboxylase. Okay, we make our oxaloacetate. Okay, and what we're going to see is we're going to use GTP here to phosphorylate this, and we're going to see our double bond here. So we're going to kick off our carbon that we just added, and it's going to come off as carbon dioxide. We're going to consume a GTP, and we're going to make this phosphoenolpyruvate. Okay, and this is known as phosphoenolpyruvate carboxykinase, or because that's really long and hard to say, PepsiK. PepsiK. All right. All right. So, all right. Sweet. Okay. So these first two steps are in the mitochondria, and then then we need to send our PepsiK out of the mitochondria into the cytosol. Okay. So we can see we can transport our lactate into the mitochondria, where we convert it to pyruvate. Pyruvate crosses there, gets acted on to form oxaloacetate and phosphoenolpyruvate, comes out that way. We can also make malate here. You can recall our aspartate malate shuttle as well too. There are also cytosolic forms of PepsiK as well. Okay, so we can see we can use malate here, cytosolic malate dehydrogenase, and cytosolic PepsiK as well. So we have multiple isoforms that are in different locations within the cells for differential regulation. So again, we can have multiple layers of regulation. All right. We can also use this oxaloacetate in the citric acid cycle if we need it there. Okay. More on that later. Okay. So I would say this first two steps in here to make phosphoenolpyruvate are big ones to know. The next steps are just the reverse. Okay, so we go back 7 steps, and eventually we get to like step 3 of glycolysis is like step 8 of gluconeogenesis. We are going to convert fructose 1,6-bisphosphate to fructose 6-phosphate. All right, so we have this fructose bisphosphatase 1. So it coordinates with phosphofructokinase and is optically regulated. So if we have high levels of ATP, we are going to slow down one direction and speed up the other direction. Same thing with NADH. All right, so we're going to use water to cleave our phosphate in this one as well too. Our hexokinase, the cognate to that is glucose 6-phosphatase. So we're going to make glucose so it can go across the glucose transporter from the liver into the bloodstream and get transported to the brain and the muscles. So again, we're looking at this in the endoplasmic reticulum, and again, I should say cleaves phosphate with water, and again, does not generate ATP. Okay. So gluconeogenesis is expensive. It costs us 4 ATPs and 2 GTPs and 2 NADHs just to make 1 glucose. Okay. But it's necessary because the brain, the nervous system, and the red blood cells only get ATP from glucose. There's only glycolysis in the nervous system and the red blood cells. Okay, so basically this is going to let us make glucose when our glycogen stores are depleted. So climbing up there and I've used up all the glycogen in my muscles, I'm going to have gluconeogenesis activated and trying to make more glucose to that. We can see this during starvation, during vigorous exercise. The other thing is we can get our glucose from amino acids, but not from fatty acids. Okay. Alrighty. So where does gluconeogenesis, where do we start from here? Well, animals can produce glucose from sugars or proteins, okay, just not lipids. Our sugars can make pyruvate, lactate, or oxaloacetate. And our proteins, we have various glucogenic amino acids. So I know if anybody has ever had to, a particular diet where they've had to monitor glucogenic compounds in there, those glucogenic proteins in there. We cannot make glucose from fatty acids. So what we do when we degrade fatty acids, we end up getting acetyl-CoA, and we can't convert acetyl-CoA to oxaloacetate. However, there are plants, yeast, and some bacteria that can do that, so they can get some glucose from fatty acids. Okay. Oops. Okay, we're getting close to being done. Okay, so let's look at our amino acids and what they can form. And the good news is you do not need to memorize this. You may need to use this at some point though. So if we look at our glucogenic amino acids based on their entry, alanine, cysteine, glycine, serine, threonine, tryptophan can all be used to make pyruvate. Arginine, glutamate, glutamine, histidine, and proline can make alpha ketoglutarate. See, asparagine and aspartate can make oxaloacetate, and so on and so forth here. Okay, I like these nice little footnotes as well too. Tryptophan, phenylalanine, and tyrosine and isoleucine are also ketogenic, but we'll get to that in a later chapter. Okay, oops, ding. Okay, let's talk very briefly about the pentose phosphate pathway. Okay, this is another fate of our glucose. Okay, so we can have our glucose 6-phosphate. We could go into glycolysis, or we can make the 6-phosphogluconate and eventually come up with ribulose 5-phosphate and make ribose 5-phosphate. And this becomes our essential for building our nucleotides and various coenzymes in our DNA and RNA. Okay, so we have an alternative pathway for glucose 6-phosphate. Part of this will rely on utilizing NADPH instead of NADH. Okay, so we're going to generate our NADPH here, and then we see our glutathione reductase combined with this as well too. But anyway, our pentose phosphate pathway utilizes NADPH instead of NADH. Also see we're going to produce some of that there. Okay, so the main products of the pentose phosphate pathway are NADPH and ribose 5-phosphate. NADPH is another awesome electron donor, just like NADH. It's required for biosynthesis of fatty acids and steroids, and oxidative damage repair will use it as well too. So again, it's another nice little electron donor. Ribose 5-phosphate, we'll see more on that later, used for the synthesis of DNA, RNA, and a lot of coenzymes. Okay. Okay, so this is the probably full pathway in here. We're not going to sweat it. I just want you all to be comfortable with looking at the components that are in here, and then we'll talk about regulation in an upcoming chapter as well too. All right. Yeah. Okay. And again, our pentose is, we are essentially, so one of the things to look at here is in glycolysis, we took our glucose and cleaved it into 2 3-carbon units. Well, here we're removing 1 carbon, okay, as carbon dioxide, to get a 5-carbon structure, our ribose 5-phosphate. Okay. We can even make xylulose. I don't know. Anyway. We have a non-oxidative phase, okay, that can regenerate glucose 6-phosphate from a ribose 5-phosphatase. Ribose 5-phosphate. And then, we can ultimately go over here with these trans-ketolases, trans-aldolases, so on and so forth to make these intermediates, but ultimately we get to glucose 6-phosphate. Okay. All right, so basically we have a reversible pathway for this as well. All right. The neat thing is there's no ATP that's used in the non-oxidative phase. We just do all these, um, you know, carbon transfers. So you can see, we make a 7-carbon member and a 3-carbon member from combining these two 5 carbons. And then we can transfer one of these carbons to make a nice little 6-carbon. We can do the same thing here, same thing here. So we're just kind of following our carbons. If we were going into great detail, this is always kind of sticks at the back of mind. This would be a good take-home question. Suppose carbon-3 were, radio labeled. Where would you find it at the end of some pathway? But on the one hand, I like that as a nice thought question. On the other hand, for undergraduate level biochemistry, I think that might be a little bit much. But I don't know. It's fun, maybe, sort of, kind of. Wait for you guys to chime in with, no, it's not fun. Don't do that. All right. One of the neat things about NADPH is it helps regulate this partitioning into either glycolysis or the pentose phosphate pathway. So if I have high levels of NADPH, then we will feed back and inhibit the pentose phosphate pathway and shunt our glucose 6-phosphate into glycolysis. On the other hand, if I have low levels of NADPH, well, I'm low on that, so I will not inhibit this. So I'll see more pentose phosphate pathway activation in there. Okay. All right, G6P dehydrogenase deficiency. Okay. So some people have a deficiency in the glucose 6-phosphate dehydrogenase. If you do a big workout, man, I tell you, my children are so loud. If you have a deficiency in this, it can be fatal. But typically, if you're taking it easy, you won't see as much there. One of the neat things, though, is you do end up with resistance to malaria through this because of this high oxidative stress in the red blood cells. Okay. We may look at a case study later on, but not anytime soon. Okay. So to summarize this chapter, yay, summary, right? We learned about glycolysis and gluconeogenesis and a little bit about the pentose phosphate pathway. Okay. So the big things here between glycolysis and gluconeogenesis, what are the differences between the two pathways? They're 7 of the 10 steps are identical. It's just that hexokinase and PFK1 as well as pyruvate kinase are all the irreversible steps. So we need irreversible steps going the opposite direction with our phosphatases and our PepsiK. Okay. I was gonna say, you guys are like, oh yeah, okay, this is not too bad, right? Right? Yeah, it's all good. It's all good, man.
Yeah, it's not that bad.
Okay. So, just as a peek ahead, I know we're not going to go too far into that. Let's make it worse. If we look at chapter 15, we now need to say, when we know we have these two similar but slightly different pathways that basically do the opposite thing. So this is where we start talking about regulation. Okay, and we start off going, how do we regulate them? And the principles are not that bad. We just look at our basic principles for biological systems. If I need energy, I want to upregulate pathways that produce energy. If I'm in a well-fed state, I have lots of energy, then I want to downregulate those energy-producing pathways and upregulate storage pathways. Okay, so we'll look at a few cases. When do we have glycolysis? When do we have gluconeogenesis? And we'll talk a little bit about the chemistry and the regulation of glycogen metabolism. Okay, so we'll bring in some studies here. We'll kind of look at maybe like diet, nutrition, and activity, well-fed state versus starved state. We can call it like preprandial and postprandial. You might see that in some biology classes, but and then we can look at extended times, extended starvation, and what happens in there as well too. Then I make it worse by saying, well, suppose we have a disease state or something in there, but I'm not gonna murder you guys with stuff. I'm basically just going to introduce this. Okay. But our principles of regulation do become ultimately very important for our overall biochemistry. So that we want to maintain that homeostasis. So again, we're trying to keep an even number of pools. We're trying to keep everything balanced. Kind of like when you have all your different classes and your different assignments, it feels like you're juggling. You got to keep them all balanced in there. Everything's balanced. Everything's good. And then something gets out of whack and you get stressed. It's like, I'm going to fail out. I should tell you my story of graduate school. My first semester of graduate school, we only had one significant course, a couple little introductory things in there too. I literally went out every like 6 nights a week and drank beer and shot pool with a friend because I just didn't need to spend that much time studying. Second semester, we added another advanced course, and as opposed to the nice, easy, slow pace of my first semester biochem, boom, the guy who started lecturing was like the micro machine guy and just talking so fast, and it was so much more information, and I had to do so much of this other stuff. I was like, oh my god, I'm going to fail out. I studied longer and better and harder than ever before at the start of that second semester because I needed to make sure I knew what I knew and what I needed to know and all the good stuff in there. So yeah, but the regulation of metabolism, it's a lot. So make time for it. Make time for it. And I'll try and make sure that we don't stress you guys out. We got to keep everything balanced. Keep everything balanced. All right. I think that's all I have today. I don't want to jump into this. I think on Wednesday we will. I may have a little assignment coming your way.
What time do I have gymnastics?
2:30. Go.
Okay.
Okay. But where am I here? Okay.